So today I'm going to talk about the specific immune response. I've looked in the book and I think the way they do it is just so awful. It makes no sense whatsoever. So in this video I'm really going to try to do it as simply as possible and explain what actually happens. Because I've just taught myself this in the process of making this video and now I know it off by heart. It's pretty easy. So here we go. The immune response happens when the pathogen has entered the body and infected some body cells. If you watch my previous video, you'll see how a pathogen is digested by macrophage and phagocyte. In order to understand this video, you'll need to understand that, so make sure you know it. It's simple. Now, there are three ways of the immune response starting, and they all revolve around antigens. Antigens are easily detected by the immune system, and all foreign antigens essentially say, I'm enemy. If you did watch my previous video, or you know in general, you know that macrophages digest pathogens. Once they've absorbed all the key nutrients from it, they will release the antigens and showcase them on the surface. This is known as antigen presentation. The macrophage is now known as an antigen presenting cell. So, the first of my three ways, antigen presentation. Also, there are cells in the body that would have been affected by this pathogen. These cells essentially signal to other cells and let them know that these cells are infected. The other way is basically finding the pathogen itself, recognising it by its antigens. Remember, all three of these ways are to do with recognising foreign antigens. Now I'm going to talk about T lymphocytes and the start of the immune response. Now, T lymphocytes have the ability to bind to antigens. This is because they have complementary receptors to the antigens on their cell surfaces. All these different types of T lymphocytes attempt to bind to the located antigen. Each of these cells wants to find out if they are the chosen one, if they have the specific receptor to this antigen. The selection and the finding of the right T lymphocyte is known as clonal selection. The T cells have now found the specific T cell that is needed for this antigen. Now these correct T cells can now divide by mitosis. After this they differentiate into three different types of cell. This process is known as clonal expansion. The cells differentiate into three types: T killer cells, T memory cells and T helper cells. The newly differentiated T killer cells go off and find other infected cells. The T killer cells then kill these cells to make sure the pathogen cannot spread any further. The other cells they differentiate into are memory cells, which essentially remember the pathogen. If this infection was to occur again, the process of destroying it would be much quicker. Now, the T helper cells have a different job. They are involved in activating the B lymphocytes. The T helper cells release cytokines. These are chemical messengers that attract B lymphocytes. I'm now going to talk about the B lymphocytes. Now, B lymphocytes are different to T lymphocytes. B lymphocytes have complementary antibodies, not complementary receptors. Much like the T lymphocytes wanted to know if they had the right receptors, the B lymphocytes want to find out if they've got the right antibodies. Is it me? Is it you? They want to know who has the right antibodies to neutralise this pathogen. Finding out who has the right antibodies is known as clonal selection, this time of the B lymphocytes. The process is exactly the same as in T lymphocytes. The correct B lymphocyte is found and they once again divide by mitosis. This is known as clonal expansion, this time of the B lymphocytes. Now, these cells can then differentiate into B memory cells and plasma cells. The B memory cells, much like the T memory cells, just float about. These memory cells provide immunological memory against the pathogen. The T killer cells hunt down the infected body cells. These plasma cells can now produce the right antibodies to actually neutralise the pathogen itself, therefore stopping the spread of infection. So just a quick diagram. At the top left you have an infected cell, in the centre you have a pathogen, and in the top right you have an antigen presentation cell. All three of these cells at the top carry foreign antigens. The T lymphocytes have complementary receptors to certain antigens. These T lymphocytes want to find the right T cell that has the right receptors for that antigen. That is clonal selection. Those cells then divide by mitosis. That is clonal expansion. They differentiate into three cells: T killer, T memory and T helper. As you can see by the diagram, the TK, the T killer cell, goes and hunts down all the infected body cells. The T helper cells send cytokines to stimulate the B cells. Now, B cells have complementary antibodies to the antigens. They have to find the right one. That's clonal selection. Once they found the right one, they expand and make more. That's clonal expansion. These cells can then differentiate into B memory cells and plasma cells. The memory cells provide the body with an immunological memory against that pathogen. Remember, the plasma cells then produce antibodies to neutralise the pathogen.